I think that.
All right.
I'll just turn it off.
Can you all hear me on Zoom now? Okay, great. Thanks, Aaron. Well, everybody on Zoom missed it, but Audra gave me a very nice introduction. Thank you, Audra.
That's why you should come to seminar. That's why.
All right, I'll be talking about some work that I think I talked about in its early stages the last time I presented this meeting, which this was pre-COVID, and we got interrupted by the fire alarm twice, maybe 3 times. So I'm hoping we do better this time. So this whole project is motivated by this premise that we talk about the gut microbiome, but it's not really one thing. Typically, very often, fecal samples are used as a proxy for the microbial content of the gut writ large. But there has been a substantial amount of work done showing that different regions of the small and large intestine, and in fact, differences in the lumen and the mucosa tissue-associated microbiota. And so these different kind of subcommunities of bacteria may be contributing to inflammatory bowel disease. In the case of this study, colitis in mice in a differential fashion. So there are microbes that are known to adhere and invade the mucosa and epithelium, including many species of E. coli. And so being able to differentiate these communities and in fact assay the microbes that are able to reside in and on the mucosal surface is really important at getting down to the immunological effects of these different communities. So the technical challenge with these mucosal communities is that we can sample the tissue, the amount of microbial DNA, the microbial DNA content of these tissues is really small compared to all the host DNA. So in this project, we wanted to, one, evaluate different approaches for doing metagenomics and metataxonomics in these difficult tissues, and also to use some novel sequencing approaches to see if we can get a much more detailed assessment of those communities. So these 3 main approaches that I'll be discussing are: 1, traditional 16S. So there have been a bunch of studies looking at these mucosal tissues. We use traditional 16S, so typically amplify 1 or 2 hypervariable regions and sequence those on a Illumina sequencer. what we'll call short reads. And this, in fact, is effective at assaying microbes in these low biomass, low microbial abundance tissues with a bunch of host DNA, because the host DNA gets completely washed out by your untold rounds of PCR. This at scale is pretty cheap, right, per base pair. The two approaches that I'm going to talk about are using this newfangled nanopore sequencing. Traditionally, nanopore sequencing can sequence much longer reads, but suffers from a higher error rate and is generally more expensive per nucleotide than your traditional Illumina sequencing. All of those things have been improving for now the 5 to 10 years we've been doing nanopore sequencing. When we first started, the error rate was 15 to 20% per base, and we get relatively modest throughputs. These days, with particular kits, we can get around 1% error. So it really has improved in both throughput and error rate over the time we've been doing this. So using these long reads, we're going to try two approaches. One is we can sequence the full length of the 16S operon instead of just a couple of variable regions. And we're also going to attempt to do whole shotgun metagenomics, even of those tissue-associated microbiota. And I'll talk about the trick that we use to do that slightly more efficiently in a little bit. But with these longer sections from each bacteria, several studies have shown that we can get much more detailed taxonomic resolution. So where traditional 16S will often get to the family or genus level, very often species and strain level resolution using these longer sequences. So some of the motivating work we did in collaboration with Janelle Arthur's lab a couple of years ago now, looking at several strains of adherent invasive and non-adherent invasive E. coli. It's kind of a proof of principle, showed, number one, these strains have dramatically different effect, dramatically different function in the gut. But if you look at your traditional, say, V1, V2, the 16S, they're essentially identical. And so by getting these longer reads, one of the things we can do is start differentiating these even closely related species, and thereby relate them to the actual function that they're performing in the gut. And our preliminary work showed that, yes, in fact, the classification accuracy was significantly better when you take the full length of the 16S compared to just a couple of variable regions. And if we go all the way out to do kind of whole genome shotgun sequencing, it's better yet. Still not perfect, although these strains are clearly genetically distinct. Every read doesn't happen to fall in a region that's perfectly discriminatory between the strains. So I'm going to take a couple of slides here to talk about the basic technical approaches to doing the full-length 16S with a neat trick, and then the shotgun sequencing, and then talk about kind of the experiment we set up to use all of this. So this full-length 16S, at first pass, you can just amplify the whole 16S operon. We're using these pretty classical probes that cover all of the variable regions and everything in between, to the tune of about 1,500 base pairs total. One of the things that we introduced and as part of the this pilot project through the CGIBD is to add, it's called UMIs, unique molecular identifiers, to each of those. And what these do is these are pseudo-random, essentially primer tails that uniquely tag individual molecules. So we can tag those molecules in the original pool of DNA and then amplify those. And this, in fact, gets us a couple of things. At first pass, we'll have multiple fragments from the same original molecule and can build sort of a synthetic consensus. This was one of the major concerns in doing this without UMIs with nanopore sequencing, is if there's a 15% error rate, it's not going to be very, very helpful, right? These aren't going to be very specific and won't have much trust in individual reads if the error rate is really high. So by building these synthetic consensus sequences, we could get that error rate much lower. Like I said, in the time we've done this project, the error rate has actually dropped a lot. But we get one extra interesting benefit from doing this tagging that, again, I'll talk about in just a little bit. So we did a little trial where we did this 16S-UMI sequencing on just an E. coli culture. We know the ground truth, so we can look at how the coverage of each of those tags, number of reads per tag, affects the residual error rate. And so in this instance, at this time, there's about a 3.5% error rate from a single read. So that's what we could expect if we didn't do the tagging. But as we get 3, 4, 5, 6 separate instances, separate reads of that same molecule, and build a consensus agreement among those, the error rate drops to where if we're over 10, it's essentially 0. This sweet spot in the end, to preserve both our accuracy and throughput, we usually pick 3. So this is getting the error rate down to just under 1%. But also, if we have to read each fragment 10, 20 times, we're correspondingly reducing our throughput. So there's a balance there between the cost and accuracy. 1% we've seen is certainly sufficient to get that gain in specificity of the taxonomic assignment. So the other trick that we use for the shotgun nanopore sequencing is something called adaptive sampling. And so this, at the time we started this project, was almost a hypothetical operation. So you can picture these pores, this blue part is, and this nanopore single molecule sequencing works such that it drives a single strand of DNA through this pore and kind of reads the electrical current that it gives off. And that's what helps deconvolve that signal into nucleotides. But what these machines can physically do is reverse the current flow on this pore on command and eject the DNA out the other side. In essence, refusing to sequence the rest of it and correspondingly kind of saving the time and effort of the pore and of the sequencer to do that. I said this was hypothetical because while the machine was capable of it, there were not good tools to predict quickly enough whether that molecule is of interest or not. And this was something that we struggled with for a while. And in fact, several of the groups came up with different and often complementary ways to quickly assess these signal, assign the sequence that's already been read to its source, and make a decision. So the way this comes into play is when we're sequencing these tissue samples that are 99% mouse DNA, we will take each read just as it begins sequencing, grab this first little snippet of read and see if it looks like it's mouse or not. And if it's mouse, we signal that pore to eject the DNA out the other way. So this is practically increasing the amount of microbial sequence that we get, so it's significantly higher than the raw abundance in the DNA sample. So we say now this can be done sort of routinely if you know what you're looking for or what you're not looking for. You can program the sequencer to do this kind of reliably, and it can reduce host DNA a bit. This has, while it's perfectly usable as described, there are some really practical challenges to increasing this. So the punchline before I get to it is that you cannot increase a sample that is 1% bacterial DNA to suddenly be 99% bacterial DNA, not particularly close to it. So if we think through how this works, it's got to start reading each fragment of DNA before deciding, and there's some minimum length it has to read to be able to assign it confidently. So what really matters for the enrichment is the average fragment length of DNA in your sample versus that ejection length. And so in this example, this average ejection length, how much did we have to read to decide it was mouse and kick it out? It was about 750 bases. But the average DNA fragment in our sample was about 2,500. So what we're getting here is just a 3-fold increase in the amount of bacterial DNA that was sequenced. All that really matters is the ratio between these two sizes. We're going to read 3 times as much sequence from every bacterial fragment as from every mouse fragment. We get a corresponding increase. Yeah.
Like 745 bases, way more than you distinguish a mouse versus a bacterium. You can do it with a small oligo, essentially, with your 16 S. So why the difference?
Yeah, for Zoom, John said 745 seems big, and it is. I misspoke. That's not the amount that it takes to assign a sequence of that length. We could do it from, I suspect, well under 100 with some confidence. The machine physically cannot flip the current that fast. So these things are flying through about 450 bases per second. So what happened was it ran for about a second, and it shot that data over to the computer. It base called it. It aligned it to the whole mouse genome, made a decision, and then sends the decision back to the sequencer. In that time, another 300 bases has gone through, and it gets stopped. So it's doing this incredibly fast, but it's just not fast enough. Yeah. And those, as far as we can tell, are fundamental limits to the actual physical hardware. So no matter how fast we make the decision, we can improve on that a lot. Yeah.
This just gives you cleaner data necessarily, but it doesn't necessarily get better than you're studying, because this is just checking for what you're sequencing, not necessarily higher.
So the throughput of the device is limited not by the number of reads, but sort of by the time and the wear and tear. So in a sense, yes, if we run this in duplicate and one is doing the selection and one is not, then we will actually get about 3 times as much bacterial DNA total by performing this procedure. So essentially the time that we've saved, the 6 seconds that it would take to sequence the rest of that mouse fragment, we can now dedicate to something else.
Because that core is now free for another.
Right. So we're reading more of each read of bacterial, but we're not actually changing the number of reads. So what this is getting us is greater sensitivity to our assignment of those reads. If what we're interested in is metataxonomics, right? Is it longer reads will give us greater sensitivity to decide what species that came from, but we're not getting more reads in a sense. So the end total of this is if we start out with 1% bacterial composition in one of these tissues, we can get it up to about 3%. Okay, but we're not doing much better than that unless we could dramatically increase the size of the bacterial DNA fragments. This is well shorter than what is ideal for nanopore sequencing and what it's capable of. I mean, these machines can sequence hundreds of thousands of bases in one go. But to get kind of a reliable and uniform sampling of the microbiota, we use a bead beating approach, which itself fragments the DNA. That gives us kind of a uniform taxonomic sampling. All right, so all of this technology, we're going to try to one, assess how well these work in an actual experimental setting, and start to see if we can see differences, one, between kind of a standard wild type and IL-10 knockout mouse across 4 different sampling sites or tissues. So we sampled from 10 wild type and 10 IL-10 knockout mice, samples from the cecal lumens, the contents of the cecum, the ileal lumen, and then tissue from both the cecum and ileum. There are ways to separate the mucosa. We didn't do that. We essentially took a cross-section, a biopsy of the full thickness of the cecal tissue and ileal tissue. But we expect that to capture any microbes that have invaded the mucosa or the epithelium.
How did you define tissue? I mean, how much do you wash it? I think there's a gray area there.
Yeah. I mean, we essentially rinse it with saline, and everything else that stays stuck, we are going to call adherent or invasive. And it would be
standardized rinsing protocol, because I guess there's probably variation in how much you shake it or how much volume you use to get off more and more bacteria.
There could have been some interpersonal variation. We didn't have one person do this alone. So I can say perhaps we didn't rigorously standardize it besides wash it off.
Okay.
Yeah. All right, let's get to results. I'm going to start with the Illumina. So we took all of these samples and we kind of put them through a normal process of Illumina 16S sequencing. These are using V1, V2 primers. And at first pass, we get kind of this very nice separation of our our genotypes. So you can see down here, these X's are IL-10 pacalmycin above are the wild type, and also this gradation by the type of sample in the tissue that was taken. And as I recall, actually, all of these groups are statistically significantly isolated from the other. We look at alpha diversity, again, still just looking at kind of the standard Illumina 16S sequencing. We get kind of an interesting pattern. Comfortingly, I suppose, we see this significant difference that is very often seen between these colitic and non-colitic mice, where there's a decrease in diversity in IL-10 versus the wild type. And this is seen in the cecal lumen. So this is the sample that is going to be closest to what we would have seen if we'd taken a fecal sample. Okay. Everything else is either in the adjacent tissue or up the digestive tract. This is kind of odd, where we see this apparent kind of huge increase in diversity in our ileal tissue. And I'll talk in a little bit about some of our theories for why that is happening. Let's see how much time I have. Okay, so we're going to move on and look at first the full-length 16S. So we only sequenced these once with UMIs, but we're going to look at them separately, essentially whether we collapse them by these UMI barcodes or not. So without collapsing them by these UMI barcodes, we're going to take this as a proxy for what if we had just not stuck UMIs on them at all, right? We're going to get the same sort of sampling we did if we had just done a straight PCR. And we see, again, kind of a nice separation. You'll see some of their ileal tissue samples fell out. Here we have 4 from the IL-10 and none from the wild type. And these essentially, they just did not amplify the full length of the 16S sequence. But other than that, we have kind of a similar separation across our genotypes and tissues, which is nice. The alpha diversity now is similar but different. We see this trend in the cecal lumen. It's not significant in this test. But again, most of our ileal tissues didn't even amplify here, and those that did have apparently lower diversity. So there's obviously something weird going on in the ileal tissue. When we look at these UMI consensus sequences, so now we've taken all of those reads and collapsed them down to where we have 3 or more reads that share the same barcode. Okay, so these should be much higher accuracy. We've dropped from 3 some odd to well down below 1% error rate, and sort of repeated this clustering. And now we've lost essentially all of our tissue samples. It's because they did not have, they were not collapsing those UMIs. Yeah.
Can I ask a question about the barcoding?
Yeah.
Quite understood. So obviously, the traditional barcoding lets you know which sample it came from, allows you to make these colors. But there's another aspect of the barcode that allows you to determine differences in the implication cycle and the error rate of the full length amplification, but I don't quite think I wrapped my head around that entirely. So
sorry for those on Zoom. I'm going to flash back a little bit.
So
yeah, so the trick is in this UMI barcode sequence. So this are essentially, they're synthesized as pseudo-random sequences. This YR structure is just to make it easier to base call for. Okay. But these are really, really long. These get ligated right on the original molecule. It's basically 2 rounds of PCR, so that each end has a unique random sequence. And then we clean that up and then go through the whole PCR. So each original molecule now has a unique random barcode on both sides.
So you can look at future amplifications against that one and see what errors there were.
We sequence the end product and we see multiple reads with the same barcode, which means they originated from the same physical molecule. Yep.
This makes more sense. And is there also a component of that barcode that tells you this was a medial tissue, this was a stool? Is there a way to resolve that as well here or not?
We do standard barcoding out here somewhere.
Okay.
So yes, yeah,
Okay.
our multiplexing for sequencing happens outside.
Okay.
So we first started looking at these. These are like 100, 200 base pair, like barcode primer structures.
Yeah. Okay.
they're kind of a beast, but yes, those
Now I
barcodes are out.
understand.
Sorry. Okay, so these all these tissue samples again dropped out, and a good chunk of many of our others luminal samples, because we didn't reach this threshold to call a consensus sequence, or didn't have enough of those to put them in this PGOA. So I think this is rarefied down to a few hundred. So any that had fewer consensus sequences than that would not appear here. All right, so the thing I promised at the beginning was that this change in approach would significantly improve our taxonomic specificity, okay? This is attempting to show the result from that whole analysis, where our traditional 16S here on the left actually seems to be doing a pretty good job deriving species 45% of the time. And the remainder are mostly genera. For some reason, we're using a normal Chain 2 assignment pipeline. The remainder it assigns to bacteria, 9%, which it says are bacteria, which, thank you, Chain. And so the full-length 16S, then, again, without collapsing those UMIs, we have a noticeably better proportion here, almost 60% assigned to the species, and it kind of goes down until we have pretty much everything after the order. The full-length 16S with UMI now kind of defies our expectations. We would think this would be, increasingly more specific, perhaps. Only better than the full-length 16S if it were the errors here that were causing it to be non-specific. But what's happening here is now this looks significantly worse than before. And my theory here, both for why the Illumina 16S is doing so well and why our full-length 16S with the UMIs are essentially analytical issues, where I think what we're capturing with the UMIs are rarer species, or we're capturing them with such precision that they in fact do not exist in our database. So we knew this was going to be a problem when we start looking at really, really detailed full-length 16S or shotgun sequencing, and that is that the existing databases, great as they are woefully inadequate to represent the full spectrum of bacteria, even that live in the laboratory mice kept here at UNC. The shotgun metagenomic sequencing in our initial analysis says we have 100% assigned at the species level. 100% is a pretty strong number, but if we actually look at it, it looks kind of like a rounding error, but the number is huge. These are extremely specific. So remember, these are on average about 2.5 kilobases long and are being anchored to one species in our database. So this, for the curious, we're effectively blasting these against the entirety of the NCBI nucleotide database. And so we're getting everything that is known, and predominantly these are extremely specific. So we want to see now, we've seen these relatively modest, I think, differences in specificity at that level. So the next question that we asked about this data is, are we seeing a shift, a difference in the taxonomic distribution depending on the approach that was used? And I apologize, I could not find a very good way to show kind of these data clustered by more than one feature. What we have is I've picked two of our mice and I'm showing our 4 different tissues here. And then also broken down, we have our full-length 16S, 16S with UMIs, and the metagenomic sequencing. And this is at the phyla level, so we see some significant variations. If we just look at our cecal lumen, these are the ones that should have highest mass, and we feel most similar to what's commonly done. We see, rather strong concordance between the 16S and the 16S collapsed by UMIs. That's good. There's this big shift in what looks like Bacteroidetes in the metagenomic shotgun sequencing. So again, this is not surprising because there are biases in 16S amplification. But seeing those kinds of shifts, if they occur across the board, is interesting to look at. When we start looking at some of these tissue samples, so it's like for 14, the cecal tissue with UMIs had essentially no consensus sequences. But then we see a lot more variation. For example, in the ileal tissue, these seem dramatically changed based on the assay approach. And just for kicks, that was the phyla. This is down, I think, just at the class level. But we see much more noticeable are these shifts in what is detected, especially in the metagenomic shotgun sequencing. So it may be that these taxa, I don't have them labeled here because there was a very long list, are going to be better sampled by the metagenomic sequencing or are missed completely via our normal 16S primers. So I showed you the PICO-A, right, which is built on beta diversity, but I think it's useful to look at these just by eye, and we can see nice differences across our tissues. So on the left is cecal lumen and the cecal tissue, ileal lumen and ileal tissue. Again, this is just at the class level, but we see these big shifts depending on which tissue was sampled. The cecal lumen and the cecal tissue are awfully similar, except for this apparent increase in, I think it's Campylobacteria here, and maybe this enrichment of undefined bacteria. But we see these kind of distinct, clearly distinct profiles depending on the tissue that was sampled. If we break these apart by genotype, I think these bars may have been shifted by my conversion of the PowerPoint here, but we can see kind of a distinct global profile between our wild type and our IL-10 mice. In particular, it looks like there's this increase in gamma proteobacteria. So why don't I just pull out our differential abundance? It tells you what actually different. Those tests are easy to run, and we did, but I'm going to explain why we're a little bit dubious of going down that road quite yet. So in our shotgun sequencing, we're getting some more data. One of the things that we looked at is just the total microbial composition versus host. Of course, in our tissues, the microbial composition by read count is very low, and in fact have a lot of the vast majority of what we sequence is host DNA. But this can give us an idea, essentially, of the relative microbial biomass in each of these samples. So as we expected in the cecal lumen, the bacterial composition is quite high. It's lower in the ileum. These often have less luminal contents also. Any of you who have ever squeezed out a mouse's ileum. And our tissues are very low. So seen another way, it looks like this. It's very high composition in the lumen. And these tissues, sort of as we expected, are down in this 1 to 2% range. So again, we can get this up to maybe 3 to 5%, but even using our fancy adaptive sampling, we just technically are not going to get a lot higher than that. Yeah.
Good question. In your previous slide.
Going back one?
Yeah.
Yeah.
Yeah. So I was a little enticed by the legend that this viruses in archaea as well. Are those not represented in your graph because they were not identified, or because, again, this was an analytical issue?
Oh, because in our various proposals, we said one of the benefits of metagenomic sequencing is we're also going to get all the viruses in archaea. And the answer is we really didn't find it.
You really didn't.
So they're plotted, but they're not there. We didn't see.
That's
The odd reader too, but like nothing that's going to show.
substantive.
Yeah, yeah, yeah. Thanks. Yeah.
So, abundance, the risk tests are due to the market, never considered that life data by high post.
Yes, thank you. I'm going to start to answer that question, I think, in just a couple of slides. So the obvious confounding factor here is our microbial abundance. Okay, so before I get to showing that explicitly, the other place we can see it again is these UMI sequences. So this is telling us something about, this is the extra benefit we get from doing this, is now we have information about the actual number of unique molecules that we're seeing there. So if we say use this process and amplify to the same concentration all of our samples, what we should see is much higher, much higher clonality of our UMI fragments from lower microbial load sequences. So we'll have 10 sequences each with 1,000 copies instead of having 1,000 unique 16S with 10 copies each. And so it's telling us something about the original, let's say diversity, but the original biomass in the sample. And so for our tissues, this is extremely low. So for doing this Illumina 16S, we use a Zymo kit that runs this in a qPCR machine, and we're getting essentially 16S absolute quantification for free during that amplification. So that's what I'm showing here, is the CT value. So recall, these are factors of 2 that as you go up, the biomass gets lower. Okay, this is a log scale on the Y. And we saw kind of exactly what we expect. The cecal lumen has this really high microbial load, and then maybe the cecal tissue or the ileal lumen. And our ileal tissue is a huge outlier here, where these had relatively extremely low microbial load. So for those of you who didn't do this quick math, that's almost a 10,000-fold difference between our average load in the cecal lumen and our ileal tissue. So we saw this weird effect of the alpha diversity. Just want to look and see, is CT values the microbial load driving difference in alpha diversity? And it certainly looks correlated, right? So this is significantly, this says our CT value is significantly associated with alpha diversity, whereas the microbial load goes down, the alpha diversity goes up. Why doesn't the alpha diversity go down as the microbial load goes down? There's some really nice work. They're not the only group to show. This is a group out of the Oregon Health and Science University who showed this really nicely, that lower biomass samples are really problematic. And that's essentially because of unavoidable contamination in the process. They did this beautiful dilution series of a community and showed that as you dilute it down, you get more and more contribution for what are apparently really, really diverse contaminant communities. There has been quite a bit of work done on this, and it's known now that the most common contributor for these contaminants is from the extraction kits. You can go and define a kit ohm for QIAGEN kits or kit ohm for Zymo kits. Unless you come up with some newfangled way of extracting DNA without using any kits or reagents in your lab, this is probably going to show up. So don't know exactly why they're so diverse or what they are, but they're there. And so you can see how these very low biomass samples that they've engineered have apparently really, really high diversity that is all coming from these contaminants. Just what they showed here is they go down this dilution series. In general, the number of unique sequences and thereby the alpha diversity is going up. Now, you might think these contaminating taxa are weird exotic things that live only in your guanidine salts, but they're not. They're really kind of familiar looking clades of bacteria, many of which also exist in the gut. And so this is hugely problematic for these really low biomass samples. We had some of these, luckily, negative controls. The important thing to do here is to make sure you sample your controls right at the beginning, right? When you dissect your mouse, you wave some swabs in the air and stick them in a tube and run them from that point on. So we run negative PCR controls like everybody does. You put water in your PCR reaction, it comes up negative, you think everything's good, but it wasn't extracted water. So we basically put water through this. I'm going to throw Zymo under the bus again, but Kaijin does the same thing. Water through the Zymo kit and get these apparently really diverse communities of bacteria coming out of thin air. So the proposed way of dealing with this is to make sure you have really robust negative controls and then somehow subtract those out of your data. Okay, the blunt force method is to take out any tax that appears in your negative controls from your data completely. That's a pretty blunt instrument, but we look at our full-length 16S and our negative controls, and again, I realize these aren't labeled because the legend is very big. But there is some significant overlap between what we see in our negative controls and what we see in our lower biomass samples. But certainly our ileal tissue samples do not look in total like our negative controls. So there is some, let's say, it incompletely explains what we're seeing in our low biomass samples, but this may certainly be contributing. So that's kind of where we leave you. I told you we are hesitant to push on to the differential abundance analysis because now we know we have biases that we haven't yet completely accounted for. And the common thing to see if we were comparing, the expected thing to see if we're comparing these samples with very different biomass is to see differential abundance showing an increase in contaminant species among our low biomass samples. Whenever we run this, we want to feel pretty confident that that's not what we're going to find. The other main benefit from the metagenomic sequencing we did that I didn't even have time to get into, but will suffer from many of the same questions, is to, instead of just assigning these to taxa, but go and assign all of our metagenomic data to, say, a protein database that we can assign the potential function of those communities directly from the shotgun sequencing data. And in the future, to develop an approach for this kind of metataxonomic metagenomic assessment that we could use for human colon biopsies that'll be robust to these kind of variations in microbial biomass. So just because a biopsy is a biopsy, but these biopsies may themselves have very different biomass. And that's a constant worry. Thank you all for your time.
Questions? Yeah, I do have a question to ask. Last slide.
Yeah.
You said that you your negative control and you subtract that by the rest of the samples.
Yeah.
So how would you do that if you have relative abundance and maybe these phyla that are actually present in your samples? Yeah. So have you ever considered like a spike and before, so you know exactly how much you could then extract that?
Yeah, those are all great ideas and things that people have thought about before. So like I said, the most blunt way to do it is to take every taxa that exists at all the negative controls and remove it. Right? But you're going to throw away signal from anything that's in your negative control that also realistically exists in your study. They said that's a very blunt approach, but maybe well-founded. There are a couple of good methods that will statistically try to remove the distribution that is seen in negative controls from your samples. Those have some evidence behind them, but I'm not totally convinced yet that that's the way. To your point about spike-ins, yes. So this is a common approach that we did not do, but basically have a positive spike-in control of known count, known concentration into your samples at the beginning, so that at least you kind of get more of an absolute quantification from the spike-in, but you can also see things that appear, especially at lower concentrations, relative to that spike-in that may help. I think you still have to answer this question. Just having a spike doesn't tell you what taxa to believe and which ones not to believe. It does help inform.
Oh, please go ahead. You go first.
So, when you are comparing these three different approaches, do you prepare all the DNAs from one single test and then divide it into three for Illumina, for 16S, and for Shotgun?
Yeah, we extracted each physical sample exactly once. So
And
the shotgun and each of the 16-ish are derived from the same DNA.
yeah, the question is that, so we all know the metagenomics or Shotgun needs tons of DNA.
Yeah.
And do you think it will be factors like affecting the total number of OTUs you got among three different approaches?
Well, yeah, definitely. And that's why you see some samples drop out completely because we didn't have enough physical DNA to do to get good metagenomic data from some. So yeah, I mean, there are concentrations that vary in every step of this process, and so it's very hard to follow through from the very beginning to the very end on exactly how many molecules we still expect going through each of these approaches, how many PCR cycles. I think, yeah, ideally, everything would be much better regularized. If I had infinite time and infinite tissue, you want to take exactly the same amount of tissue, or even normalize the tissue input based on the 16S CT values to try to normalize out the microbial load. In this case, I'm quite certain we didn't have enough material from the ileal tissue at all to reach the numbers that we were getting from minuscule amounts of the sequel. Remember, there's a 10,000 difference there. It's very hard to normalize that. I we haven't done that, and we sort of, this was incidental data that we got from the design protocol after the fact, because what that particular protocol does, it uses the CT value that comes out, she's decided to normalize the input. So in that sense, the Illumina may have normalized out some of these biomass effects, but it's clearly still those effects are very obvious in the, for example, so no, we're still very much thinking about what's the best way to do these studies and so,
Yeah, I have a lot of this work, so I have one more question.
yeah.
What other methods do you consider for the selection of Illumina content versus
What are the methods?
yeah, so basically, are you saying that you take the entire tissue and you consider that as
we're going to take the whole tissue, and we're considering that sum total of bacteria that was in the mucosal layer and perhaps it invaded with it. Of course, we're assuming there was no bacteria, right? So outside should be. But we will do that. So
I'm curious as to if whatever kits you use, if you were to take it to another lab or have it done in another lab, the kit would be the same. So I wonder what. Yeah, because it might just be my dirty pipettes that I never use filter tips with that are spraying.
yeah, the extension to that is like, do you think the ketone
yeah.
depends on A, the kit manufacturer, or B, the lab that's actually doing the experimentation? Everything.
I've done some good studies on this where they replicated in multiple labs and they took kits. There were two kinds of kits from treatment batches that were manufactured a couple months apart, and like anything else, you see effects from each of those factors.
Wow.
The biggest one seems to be the kit manufacturer and batch. But of course, yeah, there's contribution from the lab.
But I would get similar red bars if I were to take this and do it in my lab. It might not look exactly the same, but it would look more similar to that.
If you take a Zymo kit from the batch this was done in, then I hope you will get some bars.
Okay.
is there any utility in profiling kitones for some of these large population level studies?
Yeah, I think the
Because I mean,
same-
you know, what, a decade ago, everybody was convinced that this virus caused fibromyalgia. There was all this hysteria, and then discovered that XMRV was just like a contaminant hydrogen kit, you know? So, I mean, it's just, you know, just your thoughts. I'm curious to hear. I'm sure it would be an expensive endeavor, but for like HMP2 level studies, like would publishing those data alongside the actual results also be helpful?
Yeah, so I mean, to this day, a lot of studies, you said, study low biomass, where this, I think, especially in the last few years, this, but they very often don't. Making controls before the extraction. The same people who have done kind of good control studies across multiple labs have published this data and say, here's like the distribution of drugs. You can use that. You can try to take kind of the known hydrogen kit, but they found, 2 years ago in a lab across the country, it's tracking the data that helps. I don't find that very helpful, but I said there are trends. The bacteria more than the other hydrogen. I think it's not the point where it's still feel comfortable taking published hydrogen kit and just subtract.
Okay, so you think, again, you have to consider batch effects each time.
Just the negative controls with risk. Okay, the way to separate these guys.
So assuming that most of the bacteria associated with the tissue reside either in the mucus layer or maybe in the epithelial layer. I doubt there are many bacteria that go much deeper than that. Using mucosal scrapings, right, to kind of increase your bacterial biomass, enrich for bacterial DNA. I'm guessing people have looked at differences between just washed tissue and cold tissue and scraping.
You can scrape, or you can. There's a way to physically separate the mucosal layer. You can get it to secure from the epithelium. Because we were focusing on nanopore sequencing, my first thought was I need DNA, and these mice are tiny. If we were to separate the whole kind of mucosal lining of the epithelium, we still would not have had enough DNA to do the reagents. So it just becomes an absolute. It's certainly much cleaner, but we at the end of the day, we need to get something on the sequencer. We just took kind of a clip or thickness of tissue. That was sufficient. But
We use that.
yeah.
You don't.
Can you speak? I can't hear you.
Yeah, we use this great method, but we don't see like just starting with my agencies. So I was wondering, like, if it could be due to that tissue site. There's a lot of hosts. So that could be what's driving this level.
I mean, we are assuming here that none of the host units never find. So we see this effect in our 60 months, right? So I would upend the protein microbiome study to assume that the host is contributing somehow to the host DNA itself is contributing to the system. I think that's. Well, I mean, it's not coming up as microbial text. Right. These are all relative, compared to actual events. So it's a thought I don't see.
Great, this is awesome. Thank you, Jeremy.
Thank you, Audrey.
Thank you.
Thanks, everyone. Let me see if there was any questions.